appreciate it very much. Thank you for holding this hearing, very important hearing. Again, thanks for holding the hearing so we can learn more about the challenges involved with these neurodegenerative diseases such as ALS, which is a brutal disease that sadly has no known cure or any real treatment. I was particularly saddened to learn this past week of the passing of my constituent and good friend, Doug McGinnis, a combat veteran who was diagnosed with ALS over 15 years ago. He was an incredible warrior, Madam Chair. I knew him very well, and managed to fight back for many years before the disease again progressed. Unfortunately, since he received experimental treatments and that prolonged his lifetime from outside of the United States, he was barred from clinical trials here in the U.S., and ultimately he couldn't access the treatments that were effective for him, sadly. Both he, his wife, and my good friend and also constituent, Garrett Dasadi, has been at the forefront of the critical fight against this disease, and I agree we must act and do more. So my question is for Dr. Cavazzoni. We have heard and will hear the concerns from the ALS community that the heterogeneity and rareness of the disease and others, such as Huntington's disease, can complicate participation in clinical trials and access to investigational therapies. In Doug's case, these included adult stem cell treatments that proved effective for him in particular. Again, he had several treatments outside the United States. How can we better improve the drug development process so that patients with varying stages of neurodegenerative diseases are able to participate in clinical trials?
Thank you for the question. And first, let me convey how sorry I am about the passing of your constituent after a battle with ALS. I'm very sad to hear that. When it comes to the ability to access drugs despite the heterogeneity of diseases, ALS is a good example of a rare disease where we know that there are certain forms that are genetic and where we have actually been able to underpin the genetic mutation that then allows for development of very targeted drugs. On the other hand, the 90% or 85 to 90% of ALS is actually sporadic, meaning that we have not identified a specific genetic mutation or molecular underpinning. And that poses some challenges when it comes to development. On the other hand, the way we have continue to advance the development, even if we, in many instances in these diseases, we do not have a full understanding of the biology, is to work with developers and with the disease communities to identify modalities that can still allow patients to be evaluated in clinical trials. For instance, going back to the work that we have done with ALS and the guidance, we have identified a host of clinical endpoints that even without an understanding of the molecular biology of the disease or having biomarkers can allow us to adequately and quickly evaluate drugs and determine whether they can be advanced and whether the benefit and risk profile is positive. And these tools for ALS include, or these endpoints include endpoints such as function, breathing, time to tracheostomy, muscle strength, and so on, because we really want to make sure that developers can use multiple approaches to evaluating these potential therapeutics.
Madam Chair, I have a question with regard to Parkinson's disease and Alzheimer's, but I know we don't have a lot of time, so I'll have to submit them for the record. Thank you very much. Appreciate it.